In this video, we will be investigating the blood-brain barrier. My name is Martha Ugalde, and I am a student at GCU, currently taking Concepts of Human Physiology 1. What is the blood-brain barrier? The blood-brain barrier is a highly selective, semi-permeable barrier crucial for maintaining homeostasis. It is very selective, only allowing certain substances to enter the brain and prevent harmful toxins or other molecules that do not otherwise belong from crossing over. The blood-brain barrier regulates the passage of substances between blood and the central nervous system. Natural mechanisms facilitate the crossing of substances that would otherwise not be able to cross due to their size. This process involves the assistance of receptors on the blood-brain barrier. Structure and function of the blood-brain barrier. The structure and function of the blood-brain barrier consists of microvascular endothelial cells lining cerebral capillaries that penetrate brain and spinal cord of most mammals and organisms with a well-developed or functional central nervous system. Tight junctions are found between cerebral endothelial cells, and they form a highly selective diffusion barrier prohibiting most blood-borne substances from entering the brain. So on this diagram here of the blood-brain barrier, we have an astrocyte, which is very particular with the types of appendages on the cells, on the legs here for the cells. And then the blood vessel, as you can see here, has endothelial cells with tight, super tight junctions. We get a close-up of this region here. We're able to see the endothelial cells closer, the tight junctions that form between them, and then transporter proteins that are in charge of allowing certain things to cross over. Endothelial cells of the blood-brain barrier are very much different from those of the rest of the body. They have no fenestrations, they are more extensive, have tight junctions, and have no pinocytic vesicular transport. So as you can see here in section A, this is a capillary endothelial cell. These are capillary endothelial cells, and there's tight junctions in between. And this would consist of interstitial fluid. On section B, we're looking at fenestrated capillary endothelial cells. Again, there's tight junctions between each of the cells. Now we have cerebrospinal fluid next to interstitial spinal fluid. And then on section C, we have the subdural space. Here we have, again, tight junctions in between arachnoid cells, cerebrospinal fluid, and interstitial fluid on the outside. The circumventricular organs, often referred to as CVOs, are located near the ventricles of the brain. As you can see here, they're highlighted in pink. Okay, and there's one down here. The blood-brain barrier is present in all regions except for these circumventricular organs. Blood vessels around these circumventricular organs have fenestrations that permit the diffusion of blood-borne molecules across the vessel wall. So everywhere else but these organs, the blood-brain barrier is found working. Difficulties of the blood-brain barrier. One of the major disadvantages of the blood-brain barrier is a limitation it presents in delivering therapeutic drugs to the brain. So the transfer of antibodies to for neurological disorders is not possible without intervention. Approximately 0.1% of antibodies can cross the blood-brain barrier, which is medically insufficient to cure diseases like Alzheimer's. To overcome this challenge, scientists have explored ways to exploit the blood-brain barrier's natural mechanisms for crossing large molecules and engineered ways to shuttle antibodies across the blood-brain barrier. The blood-brain barrier uses specialized receptor-mediated transcytosis to allow molecules across the barrier to target a specific location or perform a specific function. Scientists have managed to leverage this mechanism to shuttle large molecules across the blood-brain barrier, which would otherwise be prohibited from crossing. This technique is now referred to as, of course, shuttling, which has opened up possibilities for treating neurological disorders more effectively, but is still a work in progress. The blood-brain barrier has several major functions that are essential for the health and proper functioning of the central nervous system. The blood-brain barrier regulates the transport of substances between the bloodstream and the CNS. It protects the central nervous system from harmful substances by restricting the entry of almost everything and anything that can cause damage to neural tissue. It assists in maintaining homeostasis by regulating ion balance and concentrations of potassium, sodium, which are critical for nerve signaling. The blood-brain barrier facilitates the removal of metabolic waste products, regulates the entry of immune cells into the brain under normal conditions, and possesses endothelial cells with unique properties such as the tight junctions that I showed earlier, which prevent the free passage of substances. These cells have elegant transport systems that actively move molecules into or out of the brain or central nervous system. It also allows for enzymes to metabolize various substances, which further contributes to the blood-brain barrier selective permeability. The integrity of the blood-brain barrier and dysfunction is implicated in various neurological disorders.